Thank you for the introduction, and thank you for the opportunity to present. So the title of the talk is Transcription of Alternative Promoters in CpG depleted region is epigenetically regulated. First of all, we define what is a reference promoter and what's an alternative promoter. So this is a schematic representation of a gene where you have a UTR coding exons, introns, and you have TSS. And the reason around the TSS is a promoter where you bind, where the pol2 transcription factors and general transcription factor binds. But when we look at ensemble or RefSeq database, we can find a gene can have a multiple transcript, and these transcripts are driven by their own promoter. As you can see here, the different transcripts are driven by different promoters. And what we use here is that the gene or the transcript with the highest expression is used as a reference promoter, and the other promoters are defined as the alternative promoters. But when we look at the sequencing data, we know that in addition to the annotated multiple transcripts, there are many transcripts that are unannotated, which people call it as unannotated cryptic or novel alternative TSS. So the question that I was interested in is how pervasive is this alternative transcription in liver cancer, and how are they regulated? So to understand this, how much of alternative transcription is used in liver cancer, we started to analyze the CADES data from human HCC patients. You have 50 tumors and the tumor adjacent tissues of these 50 patients. Then we analyze the CADES data, map this CADES data, define the tag clusters, and then we use a consensus expression to see whether these tag clusters are expressed at the population level or not. And in total, we found 42,804 consensus transcript clusters, or the TCs. Then, since we were focused on identifying the alternative promoters, we annotated these CADES tag clusters with the GenCode genes overlap with the promoters. And what we found is that we found 15,419 reference transcript or reference genes. And then these 15,000 genes had already annotated alternative promoters that were being expressed in these liver cancer samples. But we also found that many genes, many tag clusters, transcript clusters did not map in the promoter region, but many of them map in the gene body of these expressed genes, which we call as intergenic transcript clusters. And then we excluded, to find the novel alternative TSS from there, we excluded those that overlap the small RNA, that have a distinct initiations, GZ initiations motif, that overlapped enhancers, and that did not have initiations motif. They were all excluded. And in total, we found 1,031 novel alternative TSS. So in total, we found 4,083 alternative transcripts in liver cancer. And all these novel alternative transcripts do have the evidence from the RNA-seq data from the HCC patients. So what we did is now we classified this 15,419 expressed genes into single promoter and the multi-promoter, okay? And in the single promoter, you only have one TSS, but in the case of multi-promoters, you have reference and the alternative promoters. You can have one or two. And either known alternative TSS or the novel alternative TSS. And when we intersected the known and the novel, we found that some genes, some of the novel alternative TSS were already in the genes that had annotated alternative TSS. So in total, we find that out of 15,000 genes, there's one third of the expressed genes are under multiple promoter regulation. Then we did an enrichment analysis of these multi-promoter genes compared to the single promoter genes. And what we found is that the multi-promoter genes were enriched in the hepatocyte marker genes. These are the Hosida signature, which is a standard signature genes used in liver cancer to associate the survival of the patients. And they are super enriched for multi-promoter genes. Similarly, the oncogenes, transcription factor, protein kinases, they are all enriched in the multi-promoter genes compared to the single promoter. So indicating that the multi-promoters are enriched for liver and cancer-specific gene. Then we also look at the prognostic stratifications of the patients. So this is an example of a CDKN2A reference promoter, and we classify high and low expression and predict the survival probability of the patients. And we see that there's a significant difference Similarly, when you do it for the alternative promoters, you can see that there is also a significant difference. So both reference and alternative promoters can have a survival difference in terms of expression. Then we did this analysis globally across all multi-promoter genes, and what we found is that compared to the reference promoter, alternative promoters can have a better prognostic stratifications. So this indicates that liver cancer or multi-promoter genes are enriched in liver and cancer specific genes, and they have a better prognostic stratifications. So next we move on to the expression. So this is a schematic example of a beta-catenin L1 gene. Here you have a reference transcript, and here you have a reference alternative transcript. And here you have the CADES tags for reference promoter and alternative promoter. And here this is a normal liver, tumor adjacent tissue, and the tumor. And when you project this alternative promoter, and we see that the alternative promoter is not expressed in the normal liver. Okay, we have 9 normal liver samples, and then it's expressed in adjacent tissue, tumor adjacent tissue, and in the tumor. And if we look at the individual patients for these genes, this is a reference promoter and alternative promoters in the 50 adjacent tumor, and the 9 normal liver samples. And what we see is that the reference promoter is used throughout in all patients, but the alternative promoter is expressed only in some patients, and then they are overexpressed in tumor compared to the adjacent tissue. So this raises a question: how many alternative promoters are tumor specific? Or out of the 4,000 alternative promoters, how many of them are expressed only in a tumor specific manner? And to do that, we compared here whether these alternative promoters were expressed in the normal liver or not in these 9 samples. And here you have a single promoter genes, and here have multi-promoter genes. And these are the expression, the transcript clusters 3 TPM, which is the expression that is used throughout in the paper. And we find that at that level, 50% of the alternative promoters are expressed in a tumor-specific manner. They are not expressed in normal liver. And as you decrease the expression to see where you would find, whether you would see the difference. And we see that the reference promoter almost you express everything at 1 TPM. Even at this low threshold, you see that there is a significant amount of alternative promoters expressed in a tumor-specific manner. And the alternative promoters that are expressed in normal livers are specific for metabolic functions, which you would expect in the case of liver HCC patients. And the alternative promoters that were expressed in a tumor-specific manner were enriched for beta-catenin, E2F, G2M checkpoints, MYC targets. These are the pathways that are often dysregulated in tumor. So then we started to look into how does the alternative promoter impact the gene expression. Okay, so we do the differential gene expression at the promoter level, and you can see that the numbers of upregulated and downregulated promoters. And here we do the differential expression at the gene level. Basically, when we compare the expression of multiple promoters. And what we found is that at the gene level, at the promoter level, we detect much more promoters that are differentially expressed between tumor and adjacent tissue, but not so much at the gene level. And the reason is because when you start to look at the fold chains of the reference promoter and alternative promoter, we find that the alternative promoter is generally tending for upregulation, whereas the reference promoter is tending towards downregulations. So the two promoters going in opposite direction makes, then you have low resolution in detecting when you do detecting differentially expressed at the gene level, because the two promoters of the same genes are in opposite direction. Then we wanted to look into the associations with the promoters and the CpG islands, because 2/3 of the human genes have CpG islands in the promoters. And we also know that many genes have CpG islands in their gene body, or intergenic CGIs. So here we classify the system to see that we have a reference and alternative promoters and transcript. And you can find a condition where the CpG island is shared by both reference and alternative promoter, as in the case of gene as gene. The same CpG island is covered in both reference and alternative promoter. And you can have a unique cases where you have both individual cases or only in one of them, right? So we try to see now what is the distribution of the CpG island in reference and alternative promoter. And what we see is that the single promoters and the multi-promoter reference have around 80% of the promoters have CpG islands, which is expected, or which is what you would expect at the background level. But in the case of the alternative promoters, the CpG island is present only in about 50% of the promoters. But even in this case, the half of the alternative promoters are actually shared with the reference promoter, because they are, some CpGs are a few kilobases long, and they just share the same CpG island. Then we ask the other question, out of the alternative promoters that are known and that were novel, that we predicted, is there a difference? And we find that the novel alternative promoters are even more depleted for CpG island. So the new transcription, or the tumor specific activation, is happening in the non-CpG region. So that's a question, why is this happening? Because if we look into the embryonic cells, we see that the novel transcriptions are mostly happening in many of these introgenic CGI. So we try to understand why in tumor the transcription is not happening or depleted in the introgenic CGIs. So to do that, we try to look into the epigenetic regulations, histone modifications, and DNA methylations. And if we take the CG-rich region has high histone modification of S3K4ME3 in active state and S3K27ME3 in the repressed state. And when you look at the CG-rich region in the DNA methylation, that in the CG-rich region, you have low DNA methylation. And in the CG-poor region, you have high DNA methylation. This is in the normal cells. Whereas you look at in the cancer, you see that the low DNA methylation or CG-rich region, often they have high DNA methylation, and the CG-poor regions have low DNA methylation in cancer. So with this, now we try to look into both into histone modification and DNA methylation in reference and alternative promoter. And here is an example, a reference promoter, and we look for K4ME3, K27-acetyl, ME1, and K27-ME3 in the three categories based on the CpG island that is shared, unique, and no CGI. And you see that there is a very nice bidirectional K4ME3. There is a gap here around the TSS, but in the case of non-CG, there is asymmetrically only enriched in the downstream. But K27-acetyl is enriched in both directions, right, with respect to TSS. And K4ME1, when there is a CpG island, the K4ME1 peak is further away, and as the K4ME1 is as the K4 CpG island is, when you go away, the K4ME3 peak is coming closer, right? And K27ME3 is not there because these are active genes. But then when you look at the alternative promoters, you see that the large majority of them do not have CpG island, and they have a very low K4ME3, but still there is a K27-acetyl, and K4ME1 is enriched here. The K4ME1 is enriched because there is no, no very small nucleosome depleted region, and the K4ME3 that was flanking the K4ME3 is coming closer, and that's why we see a continuous trend of the K4ME3 here. So we try to look this from a different perspective, and what we did is here we take all the promoters, CpG promoters, right, reference and alternative, and we plot K4ME3, K27-acetyl, and ME1. And we see that this is the entire length of the CpG island, and there is a clear enrichment of K4ME3 and K27-acetyl across the entire body, but K4ME1 is flanking outside, right? When the K4ME3 is dropping, K4ME1 peaks. There is a peak of K4ME1 on both ends. So on the other hand, because there was no transcription in the introgenic CGI, we found that we didn't see any active histone marks in the introgenic CGIs. Then we also look at the enhancers that overlap with the CGIs. This is the CGI viewpoint and this enhancer viewpoint. And when the enhancer overlaps with the CpG island, these have the same histone profiles as the CpG island promoters, right? Indicating that the K4ME3 is enriched in the CG-rich region, irrespective of the promoter or enhancer. On the other hand, when enhancers don't have CpG island, K4ME1, ME3 is not there, and you have this unimodal kind of peak on ME1 and K27 acetyl. Then we try to look into the DNA methylation. Okay, we have a reference promoter, alternative promoters, which are again classified based on the CGI promoters and non-CGI promoters. And what you see is that this around the TSS, in the CGI, you have low DNA methylation. And as you move away from the TSS, either in downstream or upstream of the region, there is you start to see there is a global depletion of the DNA methylation in the CpG depleted region. Whereas in the case of non-CGI promoters, you start to see the DNA methylation even around the TSS because it's a low CpG region. And the same thing is also conserved in the alternative promoters in the CG and non-CG cases. And these are the individual CG probes that are differentially methylated between tumor and adjacent tissue. But when we look at the unexpressed genes, what we found is that the CpG island, in the case of unexpressed genes, there is a gain of methylation in the tumor, indicating that there is a high amount of hypermethylated probes in the CGI promoters of unexpressed genes, indicating that there is a switch in the repression from PRC2-based mechanism to DNA-based repression. Now, we again look at the DNA methylation from the CGI perspective. And now we take all the promoters, right? And we can see that the CpG islands, there is very low methylation, not much of change in the expressed genes between tumor and adjacent tumor. But as you move away, there is a change in the DNA methylation. Whereas in the case of the introgenic CGI, in the case of introgenic CGI, we now know that the introgenic CGI do not lose methylation, unlike the CG poor region. And that is the reason why there was no open chromatin and no activation, no activation of transcription or no K27 acetyl mark. That explains why the transcription did not happen in the introgenic CGI. But now we start to look into the factor that can actually explain that can explain this. So we look into this is in gene, and when the gene is transcribed, the pol2 is moving along the gene body. The pol2 will recruit the SETD2. SETD2 will recruit the S3K36 along the entire gene body. K36 will recruit DNMT3B, and this will methylate the CG in the gene body. That's why we have high DNA methylation in the gene body of active genes. Now, what we did is we look at the SETD2 in HepG2 cells, SETD2 wild type in HepG2 and SETD2 knockdown, and we profile the K36. And what we see is that when you knock down the SETD2, there is a global depletion of K36 along the gene body. And if K36 is depleted, you would expect the NMT3B recruitment will be low, and then there's a low DNA methylation in the gene body. And if low DNA methylation, there is a potential of open chromatin and pol2 binding and eventual transcription. So what we did is that now we look into the internal CpG island, right, introgenic CpG island. And what we found is that compared to the flanking region, the histone 3K36 has high affinity in the CpG island region compared to the CG poor region. Even in a knockdown, when you knock down, it still retains higher than the other. And if K36 is retained higher, this also will have the same tendency with the DNMT3B binding along the gene body, that still will have the CG rich region will have high DNMT3B binding, high K36. So there is a lot more of DNA methylation to demethylate to start the transcription. And that is the reason why there was no transcription happening in tumor in the CpG rich region. So we further went on to validate this in the patient model, and we took the TCGA HCC patients where we have SETD2 mutations and SETD2 wild type, and we compared the DNA methylation in SETD2 mutant and wild type, and we see that there is a lot of loss of methylation in the SETD2 mutant, and these methylations happen predominantly fall in the gene body over here in the SETD2 mutant. And if this SETD2 mutant now we look at the RNA-seq data for the SETD2 mutant, and we find that there's a lot of intronic reads intronic transcription happening in the SETD2 mutant that support that de novo transcription happening in this SETD2 mutant cases. So, to summarize that, alternative promoters are pervasively transcribed in liver cancer, and many of these are alternative tumor specific, which are enriched in key cancer pathways and they have better prognostic prediction. Alternative promoters are depleted for CGI. They have a lack of K4ME3. There is a dynamic DNA demethylation in the CG4 region. And the dysregulation of SETD2 alters S3K36ME3 DNA methylation across gene body, prompting alternative transcription. And I want to thank all the members from the Andersen group and the funding body and the different data source from where the data were analyzed. Thank you.
Thanks a lot, Nepal. That was really interesting. I just for clarification, so the levels of transcription of these novel promoters relative to the reference, I missed. Can you clarify how strongly are they expressed compared to the?
So they are relatively lower compared to the reference, but as you know, in here I showed, right, and sometimes it can be even higher than the reference, right? Globally, because the alternative promoters, they are not when the new alternative promoter comes, they are generally not expressed across in the entire population, but they are expressed in 30 to 40% of the population. And when you sum it, when you take an average, they appear much lower than the reference, because the reference is expressed in the entire population and the alternative is not, right? So in that sense, the alternative is indeed lower than the reference. But if you start to look at the patient level, they are very much comparable. Or not exactly 50-50, but still 70-30 level.
Do you think this is HCC specific or it's cancer specific, or there are many other contexts where this could happen?
No, I think this is a general phenomena happening in all cancer, because DNA, you are losing DNA methylation as you age or in tumor, that is known, right? And if this is a process, then you can, if you are losing DNA methylation, then there is a subsequent open chromatin, pervasive pull to binding, and and activation of transcription if you get the right transcription factor. So, but the alternative promoters that made, because here we see that the alternative promoters were liver specific. So if you take a brain tumor, you may have brain specific factors, brain specific genes that will have the alternative promoters. So it will be a context specific, but the mechanism and the phenomena, I think, is conserved across all tumor types.
And I have a question. So you mentioned at some point that the highly expressed alternative promoter have better prognostic, or it's the reverse? I don't remember.
It does not matter whether it's highly or low, but the fact that there is a second alternative promoter coming up, right? And then since this, and then this is only expressed in a subpopulation instead of rather, and then this gives a better classification of whether the expression is associated with survival or not.
Okay.
Not, but of course we separate high and low, but the absolute amount does not matter here. Okay,
so it's just the presence of
exactly
of an active promoter. And is there any somehow mechanistic reason why this activation may be related to prognosis, or is just a correlation, or there is really some mechanistic ground behind that?
I mean, for now, it's obviously a correlation, right? The activation of this, but the reason I didn't show here is that because the alternative promoter is happening in the key genes, right, in the metabolic pathways and in the key cancer-related pathway. And what I didn't show here today is when alternative promoter comes, alternative promoter attenuate the reference promoter, and the reference promoter goes down, right? So it's actually globally the reference promoter goes down when alternative promoter start to come up because and the idea is that there is a transcriptional interference.
Okay.
Right?
Mhm.
So
Yeah.
so that could be the the explanation on why this is happening, or how it can correlate, or the explanation of why there is a correlation with the survival.